Hello, I'm Jennifer Zanting. I am the biotechnology scientist at the Field Crop Development Center at Olds College. At the Field Crop Development Center, or FCDC, we breed barley and triticale varieties that are selected for growing in Western Canada. We bring seed from all over the world to Lacombe. We screen these lines in our environment here, as well as we screen for genes and new traits that the breeders could use in the breeding program. From this information, the breeders will select parents. After they cross the parents, they have to figure out how they're going to select the best lines that are created from this cross, or the best offspring. The traditional way of doing this is to just look at the crop or the line or the plant in the field. And we call this phenotyping. By using biotechnology, we can instead look at the DNA, and we call this genotyping. By using the genotype, we have many advantages. The first is that the genotype is not affected by the environment, while phenotype can be. For example, in drought conditions, a barley plant will be shorter, it may have less disease, it will likely be lower yielding. If breeders only use phenotype, they may miss the full genetic potential of that line when it's grown in a different environment. The second advantage of using genotyping is that not all traits can be seen by simply looking at a plant. Traits that are required for specific quality sometimes will require wet chemistry. This can increase the cost and the time required for screening lines. This is where the Biotechnology Lab comes in. We screen breeder lines using marker-assisted selection. By using marker-assisted selection, we can reduce the cost, increase the speed of which we can screen lines for them. It also gives us information about the genes, so we can actually mark the genes in the parents and then check for the presence of those genes in the offspring. Markers are unique pieces of DNA that can be used to identify genes or regions within the barley genome or triticale genome that are linked to specific traits. Markers can be used very effectively, and they're very similar to a page number in a book. These genes are marked within the genome, just like a page number, and then they can actually look to see if those genes are present in the barley genome. We use markers called single nucleotide polymorphisms. Single nucleotide polymorphisms are single base changes in the DNA, and it's the smallest variation that you can find in DNA. For the Biotechnology Lab, the first step to finding these markers is to get the DNA out of the plant. A small section of leaf tissue is sampled on young plants. Sample leaves is loosely folded and placed in a designated well in a 96 well plate. Each well contains a sample from a different breeding line. Samples are stored at -20 until they are frozen in liquid nitrogen and homogenized using a grinding machine to pulverize the plant tissue. The powdered tissue containing many plant cells are then broken open by adding detergent to the well. This is an important step necessary to release the DNA into the solution. We purify the DNA using a benchtop robot. Magnetic beads are added to DNA detergent solutions in each well. Then free DNA in the solution will stick to the beads. These beads with the attached DNA are then washed multiple times to remove impurities. In the final step of DNA purification, we add a specific sample solution that will cause the DNA to fall off the beads. The beads are separated from the DNA, resulting in high-quality DNA that can now be used for DNA genotyping. In order to see the the presence of a specific DNA marker, we must amplify or make billions of copies of markers that we're interested in. A process called polymerase chain reaction, or PCR, is used to amplify the DNA marker tagged with a fluorescent label. The fluorescent label is detected by the machine and thereby allowing the marker to be detected. To do this, we combine the DNA with enzymes and small synthetic pieces of DNA, which target a specific plant DNA marker. If the marker is present in the plant DNA, the PCR reaction can detect and then amplify the marker. The machine will detect the fluorescence caused by the specific marker DNA amplification. This is done in small volumes, only 3/1000ths of a milliliter. Hundreds of these samples are set up at the same time, allowing us to test hundreds of different breeding lines at the same time. As the reaction progresses, more and more fluorescence is generated, and the machine scans the plate, determining which markers is present in each plant DNA sample. Cluster analysis allows us to classify each of our unknown plant DNA samples by genotype, or the genes that they carry. This information supplies breeders about specific genes in barley or triticale lines. It also allows breeders to select for traits like disease resistance and quality. They can also start to select earlier in the breeding process and have more accurate selection. This technology allows FCDC to produce superior lines faster for producers in Alberta and Western Canada.